In this video, we'll be going to covering the topic of variation and evolution. So to summarize, basically the idea is that we've got variation amongst the same species, so that means differences, and we're going to explore how that can lead to evolution by natural selection, and then we're going to also explore how humans might use that concept to benefit ourselves in, for example, the production of food, etc. And if you're doing separate biology, then there's a little bit more about that in terms of cloning that you need to know. So we'll explore these different processes, and for the human processes, we're going to look at the benefits and disadvantages as well. So let's start with the basics, which is variation. So the word variation means differences. So we can look at variation between different species, or in this case, we're going to look at variation within the same species. So there are two main reasons that could lead to variation. First of all is our genetics. So in previous chapters, or you might have remembered covering that a gene is a section of DNA that codes for a protein, and we can have different variations of genes called alleles. And the idea is that depending on your genotype, that will determine your phenotype, which could make you look different. So for example, you can have brown eyes or blue eyes. So genetics is one reason for variation. Another reason would be the environment. So what we mean by that is that these are changes that happen to you within your lifetime. So for example, tattoos or piercings or scars that are formed from accidents, for example, these are all differences or variation between different people, for example, but that's because of the environment that causes you to have that. Okay, so for example, we can say your hair color is caused by a mixture of your genetics, which codes for your natural hair color, but you might have dyed your hair as well, and that will be a variation caused by the environment. So when it comes to looking at the variations, you want to consider, is it caused by genetics only, environment only, or is it a mixture of both? But in this case, we're going to focus on the variations that are caused by our genes. So there are many reasons for that, but one of the main reasons, for example, would be due to a random mutation that leads to this variation. And that's going to be sort of the concept we're going to base on when looking at natural selection. So the idea is that there's random mutation within the population causing variation in the population. So that means different organisms might have slightly different phenotypes or characteristics. Now, generally speaking, that's fine. Like it's absolutely normal to have differences. However, in the case of evolution or natural selection, we say that there's a change in the environment that caused some within that population to actually survive or has a higher chance of survival than the others. And it is this change in the environment that leads to natural selection. So that means nature is going to select who can survive and who dies. So like I said, we're going to focus specifically on the variation caused by your genes. So in this case, like I said previously, the mutation led some of them to have certain characteristics, and they might be better at surviving in this particular change in the environment. So they are what we call individuals that carry the favorable allele or the advantageous allele. So in an exam question, make sure that you actually specify what allele this might be. So for example, if it's bacteria, you can say it's the allele that allows antibiotic resistance, or it might be an allele that means that they are more resistant against a particular disease, for example. So you want to be specific. So those with a favorable allele could do two things. First of all, survive. Because they are able to survive, then they can reproduce. This is like a golden phrase in this topic, right? You have to say that those individuals with the favorable allele are more likely to survive and reproduce. And the idea is that they will then pass on this particular allele to the next generation, for example. And the same thing repeats over many generations. So the offspring, as they grow up, they are more likely to survive and reproduce, continuing passing on the allele. And the idea is that if this repeats over many generations, over time, this could lead to evolution, which is when the whole species essentially change to have the same favorable allele, because the ones that didn't have it would have died out. So evolution is the idea of how there's a gradual change in the species, and you can see how the variation leading to natural selection could cause this step to happen. Remember, the few steps are random mutation to start with, leading to variation. Then there's a change in the environment, be specific what that change would be. then those with the favorable allele, again, be specific there, could survive and reproduce. Sometimes they could be 2 separate marks there. They can then pass on that particular allele, and the process repeats until the whole population have the same favorable allele. Now, as you can imagine, that this process takes time because it really very much based on the environment and it happens in nature. So how can we as humans use that concept for our own benefit? So we can use something called artificial selection, or we call this selective breeding. So this is a case where the humans are going to be choosing certain particular traits that we would want. So certain traits or features or characteristics of organisms that would help us in various different ways. So that's the difference between nature selecting who's best adapted for the environment versus here in this case is human choosing what benefits us. So generally speaking is the idea of how humans choose certain organisms with the desired trait and we allow them to interbreed. We choose with amongst the offspring who has that favorable trait, and then we make them breed again until we repeat this over time until all of the offspring have that particular favorable trait that we want. It's going to be really, really helpful if you practice exam questions and see, again, particular scenarios that they can throw at you and notice how the mark scheme might change slightly. So on the whole, the steps are going to be the same, but they might be looking for very particular keywords within your answer. And again, you want to make sure that you don't just say desired traits, you want to say the actual trait that they want. So for example, individuals that make the most amount of meat. So just make sure you practice exam questions to ensure you get the best possible number of marks. So I mentioned a few things already, but let's look at the pros and cons. So the first thing that is good about selective breeding is that I can increase the resistance in crop plants. So for example, we could say increase disease resistance, pest resistance, or weather resistance in crops. So the idea is that less crop plants are going to die when, let's say, when there is a disease or pathogen spreading amongst them, but they are going to be able to survive. Another thing is that not only are they surviving and not dying, they actually make bigger crop plants. So it could be that the crop grows bigger and faster compared to just naturally existing plants, or it could be that they're making bigger fruits, for example, or bigger vegetables. And both of these together, the whole concept is about increasing crop yield. So this is a very useful phrase to use when answering exam questions. But however, it's actually better if you kind of mention those 2 points that I've mentioned first, like increasing disease, pest, or weather resistance, or making bigger fruits, making bigger crops, which ultimately both of them together leads to a higher crop yield. If you simply say in the exam that you have a higher crop yield, sometimes that can get you a mark, but in reality you could have gotten 2 marks if you mention how they increase the crop yield, which are these 2 points I've mentioned there. Having said that, it's really important that you look at the exam question and see what scenario it is that they're giving you and make sure that your answer caters to that. So let's look at some of the concerns or disadvantages. So first of all, because you are only choosing to grow individuals or breed individuals with those desired traits, and remember those traits are coded by their particular genes or alleles. So because you're only allowing certain genes to be passed on, we say that we have a decrease in genetic variation. The idea is that if the environment changes again, these individuals with those desired traits are much more likely to not be able to adapt to that environment because you are so specific about those genes for those traits that you want. Some of the other genes and alleles would be lost. And kind of linking to that is the concept of inbreeding. So that's when we say that the siblings, like the offspring in the same generation, are making more babies essentially. So the idea is that you increase the chances of the offspring inheriting a recessive allele, which means you're increasing the chance of them having an inherited disorder. And again, linking to the previous point, less genetic variation, which means they're less likely to be able to adapt under natural selection. So we kind of need to have a balance between the two. Now, selective breeding, like I said, is really useful, especially in farming. But the thing is, it takes time. right? It takes quite a few generations of this until all of the offspring definitely has the trait that you want. So there's a faster way of doing this, which is genetic engineering. So the concept of genetic engineering is about transferring a desired gene into a different organism. So we use it very specifically, perhaps in farming a lot of times, but also we use it in medicine a lot. So the example we're going to look at is about using genetic engineering to produce insulin, which is the hormone that reduces our blood glucose level. So people with diabetes, for example, or type 1 diabetes, they will need regular insulin injections. So actually a lot of the times nowadays, the insulin they inject themselves with would be made by genetic engineering. So you need to know about the process of genetic engineering if you're doing separate or combined higher. If you're doing the foundation tier, then you are not expected to know this particular process. But regardless of what tier you're taking, you need to know the pros and cons for genetic engineering. So first of all, here we've got two cells. This cell on top here, let's say that's a human cell. And inside the human cell, we've got lots of chromosomes, and here's one of them. And the red bit here is representing the insulin gene that we want to transfer. Whereas the bottom here, this is a bacterial cell, and inside it is the plasmid. So remember, plasmids are extra rings of DNA that can be found in bacterial cells. And the idea is that they don't need them to basically grow, but they provide extra DNA that could help with their survival. So one of which, for example, could be that the plasmids carry antibiotic resistant genes, and if a bacterial cell has that plasmid, then they won't be killed by the antibiotic. So in this case, for genetic engineering, we use the plasmids as our vector. So vector means that it's a chemical basically that carries the thing that we want them to carry. So you might have come across this word earlier in the specification when looking at diseases. So mosquitoes act as the vector for the malaria protist. So they're carrying the protist from one individual to another. So it means the carrier essentially. So we use the same word here, vector, to refer to the chemical that carries the desired gene. So what we essentially want to do is to take the insulin gene out from the human cell and put it into a bacterial plasmid. And the benefit of that is that if we put that plasmid back into the bacterial cell, so it can make itself very, very quickly by binary fission and essentially making lots and lots of insulin for us in a very short amount of time. So that's much faster than waiting for a human cell to be making the insulin gene. So first of all, let's say we extracted the human chromosome out, and we can use enzymes to cut the insulin gene out like this. So they are separated from the original chromosome. Same thing here, I can use the same enzyme to cut the plasmid so that it has an opening to carry the particular gene. So both of these processes use an enzyme. So after that, what we want to do is to combine the insulin gene with the plasmid. We call this the recombinant DNA. We then need to put it back into a bacterial cell. And after that, what we want to do is to put the bacteria into a culture medium or a petri dish, give it lots of nutrients and oxygen, and allow it to multiply by binary fission. So this process is kind of like cloning, right? Because binary fission is just the bacterial's way of doing mitosis by splitting itself into two once every 20 minutes. So after a while, you get lots and lots of bacteria that are all filled in the culture medium, let's say for example, like this. And what they will also do is they will start expressing that gene and making the insulin because they have the insulin gene inside them. And eventually what we then do is to extract the insulin protein and then purify it. And then after that we get lots and lots of insulin ready to be used and injected into type 1 diabetic patients. So the benefit of this process is that we no longer need to use other animal insulin, which can cause sometimes allergy problems or potential concerns of diseases as well. And of course, it sometimes would negate any potential religious concerns as well. So now that we've looked at the process, let's look at the pros and cons. So I already mentioned how we can mass produce our desired protein. We can also increase the crop yield of the genetically modified plants or crops with the same reasons as above. And like I said, make sure you specify how they might do that. They're more disease resistant or they're more weather resistant. So very similar benefits compared to selective breeding. However, the concerns are slightly different. So first concern is that the resistant gene might be able to spread to the wild type plants. So normally speaking, the GM crops or GM plants will be grown inside a very specific greenhouse, and they have very strict rules in place to make sure that you are not carrying any potential genetically modified seeds, for example, or plants out of the greenhouse. But the thing is, sometimes accidents do happen, and especially if you're growing them in a farm instead of in a greenhouse, and the plants are able to crossbreed with other species, so that gene can then be passed on to a different species. And that would result in you're giving an advantage to the wild type plants to grow better, and the concern is that they might just outcompete the other wild types, and again killing a lot of the organisms or plants because they're not very good at competing for light or water, for example. So the concern is that this would affect or potentially reduce biodiversity. And not to mention, if it's a weed plant, so it doesn't benefit us whatsoever, but if a weed plant gain that resistant gene, then we get basically super weeds. So it's going to be really, really difficult to actually kill them. There are also some concerns of potential health risks if we, as humans, eat genetically modified products. So some people have that concern. They are worried that maybe would the genetically modified genes somehow affect our own genes. Another concern would be the cost of it. So quite often the places or countries that need those seeds because they have less ideal growing conditions in their country, they may not be able to afford to buy these seeds. Whereas on the other hand, the countries that are able and can afford to buy these seeds, they themselves already have very good conditions. They don't really need it. So we have this kind of imbalance and disconnect. So there are concerns that perhaps not everyone can afford to have these expensive seeds, and that might cause concerns in terms of the food production. So keeping in mind when it comes to exam questions, just make sure you read the question carefully and see what exactly they're asking you to try to consider, and make sure that when you're looking at the actual gene, or the concerns, for example, you want to be as specific as you possibly can. So that is the overview of the topic. If you're doing just combined biology, make sure you remember that mutation leads to variation, which could lead to natural selection. And the idea is that you need to know the process of natural selection and that of selective breeding and genetic engineering, especially if you're doing a higher tier. But make sure you are also able to evaluate those two particular processes and be able to use it flexibly depending on the context of the question. However, if you're doing separates, then you want to carry on watching this video. You need to know a bit more about cloning. So as the name implies, cloning is about making genetically identical offsprings. Now, there are many different ways to do this. We want to be thinking about plant cloning first, and there are two methods of animal cloning that you need to know. So for all three of these, you need to know sort of briefly how it works and be able to compare them. So as I said, there are three methods. The first one we're going to look at would be plant cloning, and we use a method called tissue culture. So it's a very straightforward method. Plants actually have stem cells to retain the meristem in the tip of their roots and tip of their shoots, and so they retain the ability to differentiate throughout their lifetime, which makes cloning very, very easy. All you have to do is to take plant cuttings. I mean, the word kind of tells you what you got to do. You need to just snip off a part of a plant, let's say a part of the shoots, and you put it in agar, for example. And what then happens is that you make sure that the plant in the agar, you make a nutrient agar like glucose and various different things. And one of the main things you want to include would be hormones as well, plant hormones. And you might remember the plant hormone chapter. You want to include auxins in that agar to make sure that you can use it as rooting powder, and it would promote the plant growth during tissue culture. And after that, eventually they will start to grow and differentiate, growing into plantlets, which are just basically tiny, tiny plants. And once it's done, you can then transfer them into the soil and then grow them in higher amounts. So this is quite often used together with farming techniques. So especially if you genetically engineer the plant to have that particular gene that you want with the particular trait, then we can clone that particular plant by tissue culture. Now, in the case of animal cloning, there are two methods. The one that is more commonly done is embryo splitting. So what you can do, there are a mixture of different ways. You can do artificial insemination and allow the embryo to grow inside, let's say the animal's uterus, or we can do something like IVF, which we take the egg cell out and we take the sperm cell out. So for example, if you want to clone an animal with the desired traits, then you would pick the female animal and the male animal both carrying the desired trait. Again, you can use genetic engineering, technically speaking, and to make sure that the egg cell and the sperm cell carries the desired alleles that you want. Then you allow them to fuse together by fertilization, and then what you then get is a zygote. So very similar to IVF, or basically sexual reproduction. And after that, you let the zygote divide by mitosis to make a ball of cell like this. And after that, what you then do is to split the embryo up. So there are 3 small embryos, but they are all carrying the same genes, so the same cells actually, and the cells carry the same genes. So they would be genetically identical to each other. So they're genetically different from the parent, but they will be genetically identical to each other, all carrying the desired traits. So essentially what you then do is to put the embryos back into a surrogate mother. So you might put them into different surrogate mothers, allow the embryo to develop naturally in the surrogate mother's uterus until they deliver the offspring. So you can do this, for example, in cows to make sure that all of the offspring cows are genetically the same and they all make lots and lots of milk or have lots of muscles for meat. So that's embryo splitting. And finally, we have adult cell cloning, which is a less common method, and actually it's there has been lots of failures, shall we say, in adult cell cloning. It is not the most straightforward process, and basically in the past, many of the offspring that are made from adult cell cloning have lots of health problems and have died very early, have a very short lifespan. So the most famous example would be Dolly the sheep, which was the first successful case of adult cell cloning. But Dolly the sheep died like in 6 years with various different diseases as well. But nonetheless, it is a method that we can clone an animal, and the difference between adult cell cloning and embryo splitting is that we are cloning an individual that already exists. So you're cloning the donor or the adult that has donated its nucleus. So let's look at the process here. So for example, let's say in this scenario, let's say I'm going to clone a human, although in reality that is illegal, we can't do that. But just for illustration's sake, this is what we're going to have. And this particular human, it can be male or female, they have donated their body cell. So you can see that in their cell, they have the nucleus. I've colored it in red to show that this is the nucleus that we will be using. Now, on the other hand, a female has donated their egg cell. And in this case, I've colored, you got the nucleus in the center, and I've colored the cell itself in blue just to represent that we're going to be using that. Essentially what happens is that we are going to be taking the nucleus from our nuclear donor, so the target that we want to clone. We take that out, and then we put it into an empty egg cell, and because the egg cell is able to divide very quickly, and allow that particular cell to divide by mitosis into an embryo. So in this case, I've extracted the nucleus like this, and I have removed the nucleus from the egg cell. So it's an empty egg cell here. There are scientific methods to basically shock the egg cell into taking up the nucleus, making something like this. And then after that, we would again shock the cell and give it lots of nutrients to allow it to divide by mitosis to make it into an embryo. And in this case, essentially all of those cells in the embryo contain all of the genes from the nuclear donor. So therefore, this particular embryo, when it's developed into an individual, what it will be like, it's going to be identical to the original donor that we got the nucleus from. So these are 3 methods of cloning for different types of organisms. So you will notice, first of all, as I mentioned a few times, in both tissue culture and embryo splitting, the offspring and both of these processes are identical to each other. So all the planets are the same and all of these embryos are the same. However, in adult cell cloning, the offspring is identical to the nucleus donor. So that is a major difference between tissue culture and embryo splitting when compared to adult cell cloning. So in reality, at the moment, we do a lot of tissue culture and embryo splitting, especially in farming, because, like I said, we can combine it with genetic engineering to increase crop yield, for example. But the thing is, adult cell cloning is not done at the moment, or it's only done for research purposes, and it is illegal to do it on humans because of the various ethical issues and medical issues that could arise from doing so. So in particular, when it comes to concerns of using this technique on humans, people would think about designer babies. So the technique of genetic engineering is there, and so what's stopping us from basically using it on genetically engineered babies with the desired traits that we want, which has a massive ethical concern there. And again, we're combining it with cloning, that kind of doubles on the concerns of making multiple clones of humans, and there's like a big worry there. And obviously, same as before, again, we are reducing genetic variation because you're increasing the number of clones that are genetically identical to each other, and you're increasing them in the population. So again, if the environment changes, let's say crop plants are no longer able to adapt to it, then they can all be wiped out. And there you have it. That is the whole topic of variation and evolution.